Welcome friends, welcome to another video tutorial from Shomu's Biology, and here we are at the 20th lecture of Microbiology lecture series. And from now we're starting with Virology lecture. So this is the first lecture of Virology and we're going to talk about the virus structure and function in little bit details. So if you want to know about the virus, this video is just for you. Stay tuned and watch it. Okay. Now, in this lecture, we want to talk about viruses and prions. Okay, so virus-like organisms we want to talk about along with viruses. So there will be two different set of lectures for viruses. One is this one regarding the structures and properties of virus and how they infect, how they multiply, and there will be another lecture on human viruses and some very common infections and diseases caused by the human viruses and its cure. Okay, so let's begin with this first lecture on viruses and prions. The very first thing that I want to share is the search of the virus, elusive virus, and the search began not very earlier. It's until 1884. Not until that time we discovered virus, because virus is so small that we cannot know the presence of virus with the help of even light microscope. Light microscope is not enough to give us the details about the virus. So when the light microscope was invented, many organisms which we cannot see with naked eye is visible, like bacteria, protozoa, but not the viruses. Until 1884, where Louis Pasteur postulated that the rabies was caused by something which is much smaller than bacteria and which has a different way of approaching and infecting organisms and hosts at 1884. So again, Louis Pasteur started that thinking. And then 1890s, Ivanovsky and Bejernik showed a disease in tobacco was caused by virus, and then this virus, this is tobacco mosaic virus, and the disease which is very common in tobacco is actually caused by virus, which is funded by then different industries. And the work start to begin in 1890s. You can imagine, it's not that far away. So the research gradually begin from that time period, and by the time at 1950s, virology was a it's a, what I can say, multi-phase discipline. Okay, so where we have discovered a lot of different type of viruses, non-cellular particles with a definite size, shape and chemical composition. We know that they have proteins in it, they have some other chemical components in it, but they are not a true living organism, because they are not behaving the same all the time. That means once we provide them with a living cell, they behave like a living organism. Once we take away that cell, they are not behaving like a living organism. So the property and the nature of viruses depends on the presence of another living cell, which is known as the host. So whenever you find the host, the virus is living. Without the host, the virus is a non-living material. That's why you cannot even put virus in that three domain concept. Virus is totally separate. The foundations of virology, if you think about, this is a fascinating field, and you probably have heard of this term viruses, and every single layman even heard this term virus, because whenever something critical is thought about, something which is a very dangerous, which can make you sick and fall sick, example is virus. And because this term is used not only in disease for a human welfare, as well as in computer technology in this field, the computer virus. So whenever say something can destroy things, or is bad for your health of your PC, that's the virus. Non-living agents that infect all life forms. Example, phage versus animal virus. Animal viruses go and attack animal cells, while phages attack bacteria. That's why we call them bacteriophage. Viral cultivation differs from bacterial cultivation. Near about 1500 known viruses are there, and estimates to be near about 4 lakh type of viruses are there, and among them we only identify and know 1500 type. And the more we develop the electron microscopy, sorry, it allows us to visualize viruses with much up close, and we gather much more information regarding the different body component of a virus. Like in case of bacteriophage, this is a picture of a typical bacteriophage, which has a head, a collar, and the helical sheath, the tailpin, spikes, and all. So what are the general characteristics of virus? If you look at this pictures given here, all these three pictures are electron micrographs of different size and shape viruses. These are round shaped virus. This is also a rod shaped virus. This is like a very rope like coiled virus. So viruses are obligatory intracellular parasites. Intracellular means they love to be present inside the cell, and they take up nutrients from the host, and they can virtually make the host very sick and can kill it. They are filterable. The virus is Latin for the word poison. They contain DNA or RNA. They contain a protein code, okay, and the code is known as capsid, made up of capsomeres. Okay, they are of various shapes. These capsids are of various shapes. Some are enclosed by an envelope, some are without the envelope. Those one without the envelope are known as naked virus. Those with the envelope known as enveloped virus. Some viruses have spikes, which are mostly made up with proteins, and most viruses are tissue specific. So they have this tissue specificity. That means some certain virus will infect a certain type of tissue. For example, hepatitis B infects the liver tissue. Host range is determined by the specific host attachment sites and the cellular factors that a virus has. Okay. Now viruses are of different shapes and sizes. You can see this picture, and that can help you understand how much diversified these viruses are. And you can see this E. coli seems pretty big of a cell in contrast with the size of the viruses, where you can see this is the red blood cell, 10,000 nanometer in diameter. E. coli near about 3,000 to 1,000 nanometer, in this case of a rectangular rod shaped structure. While a virus, if you think about, most of them are very very small. You can see even the smallest one, bacteriophages, near about 24 nanometer. So the E. coli 3,000 by 1,000, and in contrast bacteriophage 24 nanometer only. Other than poliovirus 30 nanometer, adenovirus 90 nanometer. You can see it contains much more spikes like that. There are square viruses or like rectangular viruses like vaccinia 300 by 200 by 100. And you can see whenever we show you the different size and shapes of virus, you can see it's all kind of geometrical shapes that are available. They are polyhedrons. So they have different planes, and multiple planes are present, and we can see different kind of structures. You can see viroids, like this kind of rod like structure, tobacco mosaic virus, another rod shaped structure, very long 250 nanometer by very thin 18 nanometer thin. You can see bacteriophage T4, that is 225 nanometer, and bacteriophage M13, you can see 800 into 10 nanometer only. Rabies virus 170 to 70 nanometer. So there are different type of shapes and sizes. And you can see Ebola virus is pretty big, 970 nanometer, near about E. coli's 1000 nanometer range, but it's a totally different of a structure. So, and you know, there's a huge place of viruses and virus community that we don't know about. We only know a very fraction of that virus community. So maybe many other variety of viruses are available. Viruses, as I said, are polyhedrals. So why polyhedrals? Because of the different planes they have. They can be either this round shaped measles virus, herpes simplex virus, which is enveloped in this case, as you can see, and there are these are the shapes: poliovirus, parvovirus. Okay, polyhedral shapes that are present always. Okay, so either icosahedral and different kind of. So these are icosahedral viruses, as you can see: herpes simplex virus, poliovirus, parvovirus. Now poliovirus and parvovirus are icosahedral viruses without the capsule, so they are naked viruses, and herpes simplex is enveloped viruses. Helical virus, as you can see, tobacco mosaic virus, rabies virus, measles virus are helical, and there are complex virus which does not have any fixed geometrical structure. They have multiple structures linked. For example, bacteriophage. Head is icosahedron, and there is this the rest of the region of the body. As you can see, apart from this region, small linker is the collar, and the rest of the contractile body portion is of like a tobacco mosaic viral helical pattern. So they are composed of multiple geometrical shapes. Now the viral structure can be divided into two different types, the covering and the central core. Okay, so the covering can be of two different types. Capsid must be present in a virus, that is the protein coat inside of which the genetic material is present. Now there may or may not be envelope. Okay, if envelope is present, enveloped virus. If envelope is absent, then naked virus. Central core can consist of DNA or RNA, that is nucleic acid molecule, or they may also contain, along with that, they also contain enzymes. Not found in all viruses, but in very few cases there are enzymes present. But most of the time they only have DNA or RNA in the core. Okay, so viruses bear no resemblance to cells. They don't have any protein synthesizing machinery at all. That's why they are totally different compared to any living cell. They don't have that idea of protein synthesis, so they are not involved in the physiological properties of a cell. They don't follow those rules. So how they survive and how they replicate, how they make their more copies? They can only make more copies when they attack their host. Once they are attached with their host, they hijack the protein synthesis machinery of their host, and then they utilizes the host's enzymes for the protein synthesis. Okay, general structure. So we know the basic composition of a viral structure. Now if you think about every single component, like capsids, all viruses have capsids. As I told you earlier, protein coat that enclose all the and protect the nucleic acid of the virus, and the capsid together with the nucleic acid is known as nucleocapsid. And as you can see in both these pictures, this is the nucleic acid. You can see either it can be DNA or RNA, doesn't matter, but this is the capsid. This icosahedron structure is the capsid, both occasion. But in some occasion, maybe this icosahedron is the last layer, there is no other envelope, so it is naked virus. So naked nucleocapsid virus. And some other occasion, enveloped virus, where we have the capsid and it is covered by the envelope, and the envelope may or may not carry spikes. Okay, for few viruses the envelope protrudes some spikes out, and some other viruses there are no spike. Instead of the spike, there is the very other protein components that are present in the envelope. Okay, but generally the spike help them to interact with their host cell surface and adherence, and start to get engulfed and inserted into the host cell. Okay, and in each capsid is made of identical protein subunits known as capsomers. Okay, so the capsomers are the protein subunits which are linked with one another, stitched to one another to form the icosahedron structure. Now there are two structural capsid types available, helical and icosahedral, two different capsid types that are present. Now the helical and icosahedral, the difference between the two, if you look at here, that the helical are tobacco mosaic virus, one example, and icosahedral is the one that we studied here. Okay, poliovirus that we discussed earlier, poliovirus, herpes simplex virus, parvovirus, those one. Icosahedral contains 20 sides with 12 corners, so it's a complex geometrical shape, where helical structure is just made up with protein subunits. They are present in stack of one another like a helical coil, inside of which the genetic material is present. Okay, and there are complex virus like bacteriophage, which contains the capsid, that is the protein seed, the protein head and tail, and there's a spike and the attachment plate. All these things are present, and inside the capsid head the genetic material is present. Now if you think about the viral envelope, we have talked about the capsid. Capsid is a must for a virus because it is the primary protective envelope primary protective structure for a virus. But in some viruses there are envelope, external membrane that is present outside. So most of the animal viruses have this envelope surrounding the capsid layer itself. So occurred when the virus leaves the host cell. Generally this capsid is formed due to the modified version of the cell membrane of the host itself. So let's assume that this is the host cell, and if this is let's say the virus, and the virus is taking a part of the vesicle out, and once they bulge out from the host, a portion of the cell membrane from the host cell is captured, and that becomes the envelope. And that envelope, as we know, the host cell surface contains many receptors and other protein molecules. So that's why that envelope of the virus contains many of those cell surface protein structures known as the spikes. Okay. So made up of many identical protein subunits, it protects the genetic material, may be involved in cell entry as well as cell exit, because once they have this envelope, now next time when this virus is going to attack another host, the fusion becomes a lot easier, because now the host cell surface is a cell membrane made up with phospholipid membrane. Now this envelope is also made up with similar phospholipid component, so they can interact quite well and the fusion can be facilitated even faster. Okay. Now this is another thing. Envelope, as I said, that located outside of the capsid, and as I said, it is acquired during the viral maturation. So you think about this picture, you can see that this is the genetic material inside, capsid preventing the genetic material, okay, composed of many protein subunits known as capsomeres, and the envelope outside may or may not contain spike, but they are mostly made during the maturation. That means while the virus is going out, the virus is coming out of the host, okay, and it is facilitating this virus to attack another healthy host, okay, and the spikes help this virus to specifically bind to their target tissue and infect it. Now what are the functions of capsid and envelope? I just mentioned about it. The capsid protects the nucleic acid material from external damage, okay, and the envelope helps the virus to fuse with another healthy host, and also it has some role in tissue specificity if the envelope contains spike. Now the complex viruses like Bacteriophage or Poxviruses. Why we call them complex virus? Particularly if you look at Bacteriophage, this is Bacteriophage. Why we call it complex? Because this virus, they don't have any particular geometrical shape. They have icosahedral head, they have a very thin collar, and they have a helical sheath, and they also have a base plate. Tail fibers are attached to it, so it's very complex with multiple components. Okay, that's why we call them complex virus, because they have multiple components all together. While Poxvirus lacks the typical capsid and are covered by a dense layer of lipoprotein only. Think about it. Instead of having those complex protein subunits known as the capsomeres, they are only covered by dense layer of lipoproteins. So there is a lipoprotein layer only for Poxvirus. Now it's all about, we've talked about that envelope and capsid. But what about the core? The core part of a virus, which definitely is required. Without this part, the virus is not a virus itself, is the nucleic acid, the makeup, and the makeup of the nature of virus itself. Viral genome can be either DNA or RNA. but can never be both. It can be either DNA or RNA. Unlike animals and plants, we have DNA, RNA both. Genetic material is the DNA, but RNA is also found to carry the message. But in virus, they have either DNA or RNA, not the both. They carry genes necessary to invade the host cell, and then simply they redirect cell's activity to make new viruses. So the virus's job is not to do everything on its own. That's why virus is very simple and very lightweight and very small, because they don't need to carry proteins inside the cell. They have protein coat outside, but they don't need to carry enzymes, because what they will do, once they infect and attack a host, they will hijack the host machinery of protein synthesis, and they will produce the necessary proteins they need to get multiplied and to get spread. Number of genes varies for each type of virus, few to hundreds. Okay. Now if we compare between two different type of virus, DNA viruses usually double stranded but maybe single stranded. Now the DNA can be either circular or linear. We will talk about the example later. RNA viruses obviously are single stranded, maybe double stranded, maybe segmented into separated RNA pieces. Single stranded RNA genome ready for immediate translation are positive sense RNA. They are known as positive sense when the single stranded RNA is immediately ready for the translation process. And single stranded RNA that must be converted into a proper form are known as negative sense RNA. Because when we studied the transcription, we've talked about the sense strand of the RNA, antisense strand of the RNA, right? Coding strand, non-coding strand. But in this case, this idea is little different. In case of virus and viral infection, virus is carrying RNA. If that RNA is completely ready to be translated, we call it a positively sense or positive sense RNA. But if the RNA needs to be converted, that means, if that RNA acts as a template to make another RNA, and that another RNA is translated into proteins, then the earlier RNA is known as negative sense. That means positive sense RNA can only be converted into proteins. Negative sense RNA needs to be converted into positive sense RNA first, and then converted into proteins. Another very important thing is host range and specificity. You can see this picture animation in the left hand side here. Host range and specificity is another very important property of virus. Virus has its specific host range. It can be narrow or it can be broad. Generally, virus host range is narrower than a bacterial host range, because they can only attack a particular type of host. This is known as tissue tropism. Like in case of plants, we know gravitropism, phototropism. So similarly for viruses, we have tissue tropism. That means a virus, one particular virus, hepatitis B, infects liver. So it is specific to infect liver cells. So even if you try and check whether this virus infect other part of the cell, you try it with a cell line. If you provide liver cell, then only you will see the virus to be thriving. Okay. So again, if you like this video, hit the like button, share this video with your friends. And I believe you loved this video, so why not subscribe to our channel so that you get this kind of content continuously.